Hello and welcome back. Today we are going to discuss the neural control and coordination. We know that our body is a complex organization of different systems, and the function of these systems should be coordinated, that is adjusted to other systems. All systems are working in harmony with other systems. And who makes this harmony? Who regulates? In our body, this regulation or coordination is with the help of two systems. These two systems are one, nervous system, which is consisting of nerves. These nerves are reaching almost all important points of our body and sending messages through them in the form of electrical signals called impulses. So, through impulses, messages are brought to almost all points of our body, thereby quickly the control and coordination is brought about. But that control will be lasting only for a short period. There are a large number of events which require very quick response to our body. The control should be brought about very quickly, and that need be only short-lived. But all are not of this type. Sometimes our body requires long-term control and coordination. For that, we are having the second system, namely the endocrine system, which consists of large number of glands and scattered cells and other organs which are producing certain secretions called hormones. What are these hormones? Hormones are chemical signals. These are all chemicals. So they bring about what is called chemical coordination. This we'll be discussing in the next chapter in detail. And this hormonal regulation and control will be a bit slower when compared to that of nervous control. Control brought up by the endocrine system is a bit slower one. Why? Because the chemical is to be released to the blood, and through the blood it is reaching every cell of our body. Nervous system is sending signals to important points of the body. Signal is not reaching every cell. But here, through the blood, the chemical messenger molecule is reaching every cell. So, all cells can be controlled by the chemical coordination. Okay, it is a bit slow because it is to be transported through the blood, and though it is slow, it is long lasting. The control it brings about will be lasting for a long time. Our body requires both these two types of control and coordination. So, we are having nervous, endocrine, these two types of regulatory systems or control systems or coordinating systems, and these are the two chapters now we'll be discussing today and in the next day. Okay, so these two chapters are concerned with how our body activities are controlled and coordinated. Okay. The neural system is providing an organized network of point-to-point connection. They bring about quick coordination. Okay. Now, let us see how this coordination is brought about. Neural system consists of highly specialized cells called nerve cells or neurons. Neurons are the cells that make up the nervous system, and these neurons are capable of receiving, detecting different types of stimuli. Neurons are excitatory cells, meaning they become activated when a stimulus is applied. There are excitatory, there are highly specialized cells which are all excitatory. The neural organization is not the same in all animals. For example, in lowly organized animals, nervous system is very simple. In coelanthrates, this consists of a loose network of nerve cells which are all interconnected. You will be having one neuron, another neuron which is connected in between. So you are having a network of loose network of neurons, whereas it is better developed in insects, and vertebrates have a more developed type, and among them mammals are at the maximum level of organization for the nervous system. Okay. In human beings, the nervous system consists of two parts. What are they? The central nervous system. What is the meaning of central nervous system? Which is present on the central axis. Just like our axial skeletal system, it is occurring in the central part, which is the main processing part of the nervous system. Information from any part of the body will be brought there, and it is understood, interpreted, and sufficient orders how to respond to such conditions. They are all given from the central nervous system. This central nervous system consists of the anterior brain, which is the master processor. Okay. And it is the information processing and controlling center. And continued from that, you have the spinal cord. These two constitute the central nervous system. And arising from this brain and spinal cord, you will be having a large number of nerves. These nerves, which are arising from central nervous system, passing to all parts of the body, together is called the peripheral nervous system. It is called so because it is going to the peripheral part. So it is called peripheral nervous system. So peripheral nervous system consists of nerves of the body which are associated with the central nervous system. The peripheral nervous system has two types of nerve fibers. What are these two nerve fibers? Afferent, meaning those which are all giving message to the central nervous system, bringing information to the central nervous system. They are also called sensory, which will be bringing all messages from the different parts of the body to the central nervous system. And those nerves which are taking away messages of the central nervous system to different parts of the body, they are all called efferent. Efferent is also called motor. These motor neurons are taking information from central nervous system to different parts of the body. And this peripheral nervous system can be divided into two parts. What are they? So peripheral nervous system consists of two types of neurons. And this peripheral nervous system has two parts. What are they? Let us see. One, somatic. What is somatic? Somatic nervous system will be under our will, under our control. It is voluntary. So, all informations to our voluntary organs, like the skeletal muscles, will be carried by the somatic neural system. Whereas the second one is autonomous. Autonomous is involuntary, which is not under our will. So you cannot control by simply wishing, let this action be happen. Just by wishing, this action will never occur because it is not under our will. It is called autonomous. This autonomous system will be controlling the internal activities. The autonomous nervous system will be responsible for involuntary activities, which are all again divided into two types, namely sympathetic and parasympathetic systems. Sympathetic and parasympathetic. These two are constituting the involuntary autonomous. Okay. So we are having sympathetic and parasympathetic, which are all antagonistic in action. When sympathetic system tries to increase the rate of a reaction or increase the working of a system, the other one will try to decrease. That is how the normalcy is always maintained. These two are all antagonistic in action and brings about the homeostasis, that is maintenance of a constant and steady working of these systems. Now, visceral nervous system is a part of the peripheral nervous system, which comprises nerves, fibers, ganglia, plexus, by which impulses travel from the central nervous system to the viscera and from viscera to the central nervous system. All such systems, elements, which are all responsible for carrying of messages or impulses from the central nervous system to the peripheral organs and from peripheral organs to the central system, together is called visceral nervous system. Visceral nervous system. We are having the components like nerves, fibers, ganglia, and plexus that are forming the visceral nervous system. Okay. So this is the classification of our nervous system. So central nervous system, peripheral nervous system. Okay. Peripheral nervous system divided into somatic and autonomous. Autonomous divided into sympathetic, parasympathetic. And visceral nervous system consists of all the elements, comprise the whole complex of elements which will be responsible for the transfer of impulses from central nervous system to viscera and from viscera to the central nervous system. Okay. Now, we will be discussing one by one. Neuron is the basic unit of nervous system, which is excitable. Remember, the entire nervous system is developing from ectoderm. Ectoderm give rise to the nervous system. And there are two types of cells that make up the nervous tissue. There are neurons and glia cells. What is a neuron? Neuron is an excitatory which can be activated, which can also conduct signals. That's why it is called neuron. Okay. It is excitable. It has three components. The main part is the cyton, which is also called the cell body. It is called cyton. Here you will be having the nucleus of that cell. Surrounding that you will be having a mass of cytoplasm, and that cytoplasm will be having certain stainable structures, granules called Nissels granules. Nissels granules are actually rough type of endoplasmic reticulum plus ribosomes. Okay. This cyton will be bearing two types of process. One type is smaller in size but large in number. They are called dendrons. These dendrons end up in small branches called dendrites. Dendrons bring information to the cyton. So we say that they are all afferent in function. Afferent means bringing. Now, the second process will be a long one, which is only one in number. So it is longer, but only one in number. It is called axon. The axon will be ending in branches called axon endings or axon terminals. At the end of this axon terminal, you will be having a swelling called synaptic knob. Inside the synaptic knob, you will be having certain vesicles called synaptic vesicles that will be helping in transferring impulses from one neuron to the next neuron. Okay. This axon is having a membrane called axolemma. And inside, you will be having the cytoplasm called axoplasm. Clear? In axon, you will be having no Nissels granules. Nissels granules are present only in dendrons and cyton. They are absent from axon and axon hillock. What is axon hillock? This axon is arising, starting from a small conical structure. This is called axon hillock. In axon hillock and axon, the Nissels granules are absent. This is the first type of cell, which is called neuron, which is excitable, which can become activated, which can be activated, and which can conduct impulses through them. The second type is called glia cells. Glia cells are constituting more than 50%. These glia cells are not at all excitable. They cannot conduct signals through them. This glia means blue. are helping in grouping, bundling of axons and help them in covering them, in protecting them, in nourishing them, in providing the favorable environment for their working, etc. Such cells are called glia cells. Okay. Now, there are different types of glia cells. Glia cells can be classified into microglia, meaning small. They are small in size but are all having pseudopodial-like structure by which they will be taking and destroying large sized solid particles. So they are all macrophagic in function. The second type is macroglia. They are larger in size. There are so many different types of macroglia like astrocytes, ependyma cells. Ependyma cells are just like epithelial cells lining the cavities of the nervous system. Then oligodendrocytes. Oligodendrocytes are covering the axons in the central nervous system, whereas the axons in the peripheral nervous system are covered by another type of cell called Schwann cell. These Schwann cells are cells that are covering the axon. Sometimes these Schwann cells will be, suppose this is the axon in cross section. Schwann cells will be remaining along the axon on its side. Okay. Whereas in others, this will be forming a concentric covering. What is concentric covering? Let us see. This cell will be making the cell is making several layers around the axon and thus forming a thick covering. Such a thick covering formed by the concentric covering of Schwann cell is called myelin sheath because the walls are very rich in sphingomyelin, a phospholipid. Sphingomyelin. Since it is made up of sphingomyelin, we call it as myelin sheath. Thus, myelin sheath will be insulating a neuron. When myelin sheath is formed, the axon is said to be myelinated. The Schwann cells are arranged one after another. Okay, that means, suppose this is the axon, one Schwann cell, next Schwann cell. In between the two Schwann cells, there is a small area which is called node of Ranvier. Node of Ranvier. In a myelinated axon, impulse will be jumping from one node to the next. So impulses are conducted at a faster rate, very quickly, because impulses are jumping from one node to the next node. That way of conduction is called saltatory conduction. Saltation means jumping. Okay. Those which are having myelin sheath is called myelinated. These myelinated neurons are thus conducting impulses very quickly. And in non-myelinated, which is having no such myelin sheath, Schwann cell is present, but Schwann cell is not forming any myelin sheath. Clear? The non-myelinated or unmyelinated nerve fiber is enclosed by a Schwann cell, but that doesn't form a myelin sheath. It is commonly seen in autonomous and somatic nervous system, that is the peripheral nervous system. Is that clear? Now, let us see how we are all having the nervous system working. Neurons are excitable, okay, and it is the axon which are all mainly involved in creation and conduction of impulse, because axon is the longer one. Okay. When you apply a stimulus, a neuron become excited. Why it become excited? Let us see. Just like any other cell, an axon will be having two types of transportation occurring always across its membrane. They are active transport and passive transport. Active transportation is with the help of certain enzyme complexes which are present on the surface. It is actively transporting, meaning it is transporting ions with the help of energy. So it is active, and so it is fast and even against concentration gradient. The most important type of active transportation mechanism is the sodium potassium pump. Why it is called a pump? Just like a water pump, it is conducting ions from a low concentration to a high concentration, that is against concentration gradients. This is an enzyme system present in cells which is conducting sodium ions to outside and potassium ions to inside. It is a double pump. It can conduct sodium and potassium in two different directions. So it is also a antiport. With one molecule of ATP, it can conduct two potassium. It can pump two potassium inward and three sodium outward. Okay. It is against the concentration gradient. Why I said that it is against concentration gradient? Sodium is almost 10 times more on the outside, and potassium is almost 30 to 40 times more inside. Still, sodium is pumped out and potassium is pumped in. That is why I told you that it is a pump. It is against a concentration gradient. Okay. This is continuously working. So, when a neuron is at rest, it is spending energy for what? For the working of sodium potassium pump. As a result of that, you will be having the sodium more on the outside and potassium more on inside. That is the active transportation mechanism. Now, in the membrane, you are having some proteins which are porous, hollow tubular proteins, which are all having a channel through which specifically a particular ion can diffuse. This is passive transportation with the help of ion channels. These ion channels are proteins which are present within the thickness of the membrane, which is having a cavity through which specifically a particular type of ion will be transported. You are having sodium ion channels and potassium ion channels. You will be also having so many other ion channels, but we are all interested only in these two. Okay, because they are all helping in the characteristic maintenance of a potential in the membrane. When membrane is not conducting any impulse, membrane is impermeable to sodium. Why sodium is not permitted? Because the sodium ion channel through which they should come is closed. Whereas potassium ion channel, that is open. So sodium ion channel closed, potassium ion channel open. So these ion channels are not allowing passage of materials always, because there is a gate that regulates the flow of ions. So we call it as gated ion channels. And these gates are closed and open based upon the voltage or potential difference. So it's called voltage gated ion channels. Sodium, potassium, voltage gated ion channels are regulated by the potential differences. So when the membrane is at rest, not conducting any signal, when the neuron is at rest, what is the condition? Sodium potassium pump is working. Sodium ion channel is closed. Potassium ion channel is open. So that the potassium which is more on the inside diffuses out. So potassium is continuously flowing. So the membrane is freely permeable to potassium and impermeable to sodium. Is that clear? So already there is positive charge sodium outside. Now this potassium is also going out. Outflux of potassium causes the positive charges to get accumulated on the outside. Positive charge ions are more on the outside than on inside. What is in the inside? You are having exoplasm. Exoplasm is having proteins which are having amino acids. Amino acids are negatively charged. So we are having the membrane having positive charges concentrated at the outside, negative charges on the inside. We call it as polarized. So you will be having positive charges on the outside, negative charges on the inside. This situation is called polarized. So the membrane is not at all conducting an impulse, it is polarized. And there is a potential developed in between the outside and inside. That potential is called resting potential. This resting potential is minus 70 millivolt. 1/1000 of a volt is millivolt. The voltage developed is so small, it is in only millivolt. Minus sign shows that inside is negative. Okay, this is the condition when the neuron is not conducting any signals, it is at rest. Now you apply a stimulus. It can be either a physical, a chemical, a biological, or even an electrical signal. Suppose you apply a signal or a stimulus to that neuron. I am applying a stimulus to this polarized part. Immediately, what happens is that for a very short period, sodium potassium pump stops working and immediately starts again. And under the influence of that stimulus, the sodium ion channel is now open. Sodium ion channel is now opened. When the sodium ion channel is open, the potassium ion channel is closed. Its permeability decreases. So what happens? Potassium is not going out. And sodium is coming in. When sodium ion channels are open, sodium is coming in. When sodium is coming in and potassium is not going out, what happens? The inside become immediately positive charge. It become positive charge. Why it become positive charge? Because now sodium is here, but sodium is coming in because you are having so much of sodium, 10 times more sodium on the outside. So its concentration is very high. So from a high concentrated outside to less concentrated inside, sodium is coming in. Potassium is not also going out. When positive charges accumulate inside, comparatively, the outside is comparatively, it is negative charged. So it is comparatively negative charge. So what happened? Originally it was polarized. Now that polarized condition is changed. It become depolarized. So the polarized condition is now changed as depolarized one because that polarity is removed. D means removal. And this depolarization will be causing two important things. What are they? One, it will cause the opened sodium ion channel to become closed. It causes this sodium ion channel to be closed. Now this channel automatically become closed. And all the sodium that came in will be pumped out. So again, sodium is pumped out. Potassium ion channel is now open because the sodium ion channel has closed. Potassium ion channel is now opening, and through that potassium is starting to move out. That is number one. Two, it will induce the next point. It will induce the next point. How? It will cause the sodium ion channel in the next point to open and potassium ion channel to be closed in the next point. The potassium ion channel is closed and the sodium ion channel is opened. So what happens? The next point become permissible to sodium. So there will be influx of sodium. Originally, you are having positive charges accumulate in the first point. The next point is a depolarized one inside. The next point was depolarized. And since both positive and negative charges are coming close together, there will be a flow of charges from depolarized to the polarized next point. Charges will flow. Always charges will flow from positive to negative. So positive in depolarized, negative in polarized. So charges will flow inside from a depolarized to polarized. And to complete the circuit from the polarized to depolarized, positive charges will flow. Thus, the next point become positive inside and negative outside. Because that cause the sodium ion channel to open and potassium ion channel to close. So the next point become positive inside and negative outside. That means by the time this original point where I have applied the stimulus has come back, it has now become negative. It has become negative inside. Why it has become negative? Because all the sodium is pumped out and potassium is also moving. So again it is coming back to the original condition, that is polarized again. It is called repolarized. By the time repolarization occurs, the next point has become depolarized. This depolarization will induce the next point to become depolarized, and there is a flow of charges, and again this second point become repolarized again, come back to the original situation. So there is alternate depolarization and repolarization occurring in every point of the axon. So these points become depolarized, repolarized, depolarized, repolarized. And thus, in the form of a wave, the cyclical event of depolarization and repolarization travel through the axon. This cyclical event of depolarization followed by repolarization is called action potential. During action potential, during depolarization, inside is becoming positive and the voltage is plus 30 millivolt. Plus 30 millivolt. It immediately come back to the original polarized condition. Thus, in the form of a cyclical event, in the form of a wave, depolarization and repolarization will be passing along the axon. This is called the impulse. This impulse will be traveling till it reaches the axon end. At the axon end, let us see what is happening. In the axon end, you are having certain axon terminals with synapses, synaptic knob. These synaptic knobs are swellings at the tip of the axons. Inside that, there is a synaptic vesicle. Synapse is a junction between either two neurons or a neuron and a muscle. Junction between neuron and muscle is called motor end plate or neuromyonal junction. Whereas the synapse between two neurons is called interneuronal. In interneuronal junction, see, this is the axon terminals which are having the synaptic knobs. The synaptic knobs are in contact with the dendrons of the next one. So actually, the junction is between axon terminal of one neuron that brings information to that synapse and the dendron of the next one. So this functional junction is a synapse. Why I say that it is a functional junction? Because actually there is no contact, there is no touch of the two membranes of these two neurons. In between, you are having a small gap called synaptic cleft. You are having a small gap called synaptic cleft, which separates these two membranes. Okay. In the axon terminal, impulses are reaching, so that there will be a change in permeability. As a result of that, calcium from surrounding medium, surrounding fluid enter into, and when calcium concentration increases, the synaptic vesicles will be coming and fusing with the membrane of the axon, which is called presynaptic membrane, because it is lying ahead of the synaptic cleft. So it is called presynaptic. And the membrane of the dendron, which is called postsynaptic membrane. The vesicle, after fusing with the presynaptic membrane, will release certain chemicals they store. This chemical is called neurotransmitter. There are so many different types of neurotransmitters. Adrenaline or epinephrine, noradrenaline, acetylcholine, etc. The common type of neurotransmitters. So this neurotransmitter is released into synaptic cleft, and these vesicles diffuse across the synaptic, the chemicals will diffuse across the synaptic cleft and will fall on the receptors. They will fall on the receptors which are present on the postsynaptic membrane. What is this postsynaptic membrane? Postsynaptic membrane is the membrane of the dendron. Okay. Now, on receiving that neurotransmitter, the receptors become active and it brings about permeability changes, more sodium enter in, and thus that membrane become depolarized. When depolarization occurs, immediately repolarization will be occurring, and thus an action potential is generated here also, and that action potential is transmitted through the dendron to the cyton, and from cyton through the axon till it reaches the axon terminal. This is repeated. This is how we will be having impulses transmitted across synapses. We are having two types of synapses. We have seen that here the signal is traveling or crossing that gap with the help of that chemical neurotransmitter. Such a synapse is called chemical synapse. It is called chemical synapse. We are having more chemical synapses in our body. Second type of synapse is called electrical synapses, which are very rare in our system. In electrical synapses, the pre and the postsynaptic membranes are very close, or they may be even touching each other. The synaptic cleft is so narrow, so small, or even the two membranes may be meeting, so that there is no need of a chemical. The signal can cross as if it is passing through the membrane because the gap is so small. Or it can jump from presynaptic to postsynaptic. Such a type of synapse is called electrical synapses. Electrical synapses. So, at a chemical synapse, we are having the pre and post synaptic neurons separated by a fluid-filled space called synaptic cleft. Here, the synaptic cleft is very narrow or even absent. The membrane will be thus conducting signals very quickly across an electrical synapse. Clear? coming to the central nervous system. It consists of our brain and the spinal cord. Brain is the main processor, central processor, which is the most important part of the nervous system, which is protected by means of three coverings called meninges. Outermost meninge is a very thick fibrous covering. Okay. These meninges are right from outside. Dura matter, meaning tough mother, outermost. The middle one is very thin. So, just like the spider web, arachnoid membrane. So, third inner one is pia matter. Pia matter. So, dura matter, arachnoid membrane, and pia matter. These membranes will protect our brain from shocks, jerks, etc. Brain is also protected by our skull, which are very hard, and is protecting brain well. Okay. Brain is also concerned with the process of vision, hearing, speech, memory, intelligence, emotions, thought, etc. It is also responsible for the control of or understanding of the thermoregulation, hunger, thirst, and our body activities, daily body activities, circadian rhythms. Okay, so we are having the three membranes that cover and protect. Cranial meninges, they are also called meninges. Okay, outer dura mater, middle arachnoid, and inner pia mater. Brain has three regions: forebrain, midbrain, hindbrain. Prosencephalon, mesencephalon, rhombencephalon. That is the other terms for these three parts of the brain. Okay, this is the sagittal section of the brain. If you are taking a section of the brain, so as to divide it into a right half and a left half. Forebrain is the largest part. It has three regions in it, namely a cerebral hemisphere. This cerebral hemisphere, right, is called hemisphere, half sphere like, two halves, which are all joined together, connected together by a track of nerve fibers called corpus callosum. Corpus callosum is connecting the right and the left halves of the cerebral hemisphere. Okay, this cerebral hemisphere is having gray matter, that is neurons on the outer side, and white matter on the inside. White matter is mainly because of the myelinated axons. So, mostly nerve cells are in the cortex, but their axons are forming bundles. Large number of them are present in the medulla that gives white color. So, it's called white matter. The cerebral hemisphere is overdeveloped. In mammals, it is covering other parts of the brain. Okay. In man, it has different lobes, mainly a frontal lobe. This is the frontal lobe, which is the seat of intelligence, reasoning, thinking, etc. This is the frontal lobe. This is the parietal lobe. Just behind that, you are having the parietal lobe, which is sensory and motor. Large areas of these are both sensory and motor, and are forming what is called association areas. They are responsible for complex functions like intersensory associations, communications, memory, etc. So some areas are sensory, some areas are motor, and large areas are association areas. Okay. Then you are having a pair of temporal lobes. Temporal lobes are concerned with hearing. See, we hear sound, and that sound is interpreted. That is why we understand what is the meaning of that sound, etc. All are under the control of the temporal. And in the posterior, you are having an occipital segment, which is concerned with vision. With that, we understand the shape, we understand the color, we recognize people, we recognize things, etc. So, visual part. Then, you are having the medulla, we are having the diencephalon, which is the second part. This second part, diencephalon, is the posterior-most part. But here, the diencephalon is completely covered over. It is completely covered over by the overdeveloped cerebral hemisphere. This is the diencephalon. The roof is called thalamus. The thalamus is acting as the key communication center. Thalamus is the key communication center. Information from any part of the body will be recognized, and based upon their importance, they are distributed to the concerned region of the cerebral cortex. The floor is called hypothalamus. This hypothalamus is having a number of important functions. One, it is the thermoregulator of our body. Thermoregulator that maintains our temperature. It is this part which is controlling hunger, thirst, etc. And large number of these cells in this area are secretory. They produce secretions. And these neurosecretory cells are producing the two hormones mainly called oxytocin and vasopressin, which are all stored in the posterior lobe of the pituitary. So, though it is a very small part, it is playing a very important role, hypothalamus. In the diencephalon, you are having a cavity, which is the third ventricle of the brain. And the cerebral hemispheres are having small slit-like chambers, space inside them, called lateral ventricles. Lateral ventricles are open into the third ventricle by means of a small opening called foramen of Monro. Actually, we are also having an olfactory lobe also. But that olfactory lobe is at the lower side here. Olfactory, that is concerned with smelling. Okay. Now, in the deeper part, connected with the cortex and with the hindbrain posteriorly, you have a characteristic fork-like structure called limbic system. It is called limbic system because it is in the form of a fork or a limb or a lip. So it's called limbic system. The anterior part of that limbic system is having a structure called amygdala, which is the defense castle, which is concerned with rage, anger, that protect you from the attack by the enemies. Okay. The Second part is having the shape of, roughly having the shape of the seahorse, hippocampus. So it's called hippocampus, and it is the site of long-term memory and smell. So this limbic system is having amygdala, hippocampus, and the posterior one is septum, which is concerned with sexual behavior and emotional reactions like excitement, pleasure, rage, fear, etc., motivation. All these are parts of the limbic system. So this limbic system is present on the inner part of the cerebral hemisphere, which is a complex structure having the shape of a limb or a lip. So it's called limbic system. So we are having in the forebrain mainly three structures. One, olfactory lobe, cerebral hemisphere, diencephalon. The roof of the diencephalon is called thalamus, floor is called hypothalamus. Arising from the hypothalamus is a small finger-like structure called pituitary, which is an endocrine master gland. Similarly, arising from the roof of the you will be having another small endocrine structure called pineal organ. We'll be studying about the pineal organ, hypothalamus in detail in the next chapter. These are all the endocrine parts of the brain. Hypothalamus on the floor and the pineal organ here. Okay. Now coming to the middle part, midbrain. It is located between the thalamus, hypothalamus of the forebrain, that is diencephalon, and pons of the hindbrain. It is having 4 lobes. They form what is called corpora quadrigemina. Quadri means 4. It consists of 4 lobes. The 2 upper lobes are called superior colliculi. For rounded swellings, lobes, and inferior colliculi. Superior colliculi is concerned with reflex actions, focusing of the eye, movement of the eye, etc., quick movement of the eyes. Inferior colliculi is concerned with hearing reflexes. So this is present on the dorsal side, and on the ventral side there is a structure called, see, this is the superior colliculi, inferior colliculi. Through the center passes a central canal. It is called iter or aqueductus that connects the third ventricle that is present within the diencephalon and the fourth ventricle present within the hindbrain. And ventrally, you are having a mass of nerve fibers. Okay. It is called crura cerebri. Crura cerebri. So, superior colliculi, inferior colliculi together constitute the corpora quadrigemina. We are having 4 lobes. Whereas in frog, we are having only 2 lobes, that is mainly concerned with vision. So, we call it as optic lobes. Now, coming to the hindbrain. Hindbrain consists of mainly a pons, cerebellum, and medulla oblongata. Pons is on the ventral side. It is a bridge that contains axons that connect different parts of the forebrain with the hindbrain. Cerebellum are small structure similar to that of the cerebrum. That is why it is called cerebellum. Cerebellum means small cerebrum. It is having a number of gyres and grooves, just like our cerebral hemisphere is having. The cerebral hemisphere is having a number of grooves and gyres, so that the surface area is increased. That is why it is capable of accommodating a very large number of neurons. That is the reason why we are all highly intelligent. This type of grooves and gyres are also present. Here also, gray matter is out and white matter is inside. This white matter is divided into a number of branches of a tree. So, it is called arbor vitae. Arbor vitae means tree of life. Tree of life. Cerebellum are involuntary action. All the involuntary activities are coordinated by the cerebellum. Cerebellum is having no ventricle. Medulla oblongata is the posteriormost part, which contains centers for respiration, that control respiratory activities, digestive activities, and cardiac activities. So, it is this part that controls vital, important internal functions or internal structures and systems like respiratory system, digestive system, and circulatory system. We are having respiratory center, cardiac center, etc. in this part. Medulla contains center which control respiration, cardiovascular reflexes, and gastric secretion. Now, the midbrain, pons, and medulla oblongata, these three parts together will form what is called the brainstem. It is that brainstem that forms the connection between brain and spinal cord. The connection between the brain and spinal cord is through the brainstem. So that is the structure of the brain. Okay. Now, what about reflex actions? Our body is having an inbuilt mechanism to protect itself from emergency situations. These are involuntary. That is, at the time of that action, we don't know it. Only after the action is over, we come to aware that this action had occurred. Such involuntary, stereotyped, quick and sudden actions to protect our body from emergencies is called reflex action. When you touch a hot object, unknowingly, immediately we withdraw our hand. Similarly, when a knee hammer is hitting on the knee, immediately we will be pulling our legs. These are all typical examples for reflex action. So, without our conscious effort or thought, we make such actions always to protect ourselves. These actions are called reflex action. And the pathway through which the neurons are conducting the signals to bring about a reflex action is called a reflex arc. So, there will be a sensory organ that receives that sense. So, for example, when an insect is coming towards eye, it is the eye that is receiving that stimuli. Okay, so that is the receptor organ. Then the signal from that receptor organ is taken to the central nervous system through an afferent. Afferent is sensory. Sensory neuron, it reaches the central nervous system, and the message is never relayed to the higher centers of the nervous system because the situation is an emergency one. There is no time. So immediately the response should come to the body. So the central nervous system will be having what is called an interneuron. That central nervous system, having the interneuron part, will immediately give sufficient motor informations which are carried through the efferent pathway, motor pathway, and is brought to the muscle or gland where these orders are effected. That is what is called motor or effector organ. So immediately we may withdraw our hand or shut our eyes. Okay. So these muscles or glands are the effector organs or motor organs. So all these neuronal elements that are essential for bringing a reflex action is called a reflex arc. Reflex actions may be either simple reflexes, which is unlearned, or learned reflex actions, which is called conditioned reflex. Anyhow, for a reflex action, there must be a central nervous system part. Okay, signals are brought to that central nervous system and orders are also coming from that central nervous system. If that element is the lower centers of the brain, then the reflex action is called cranial reflex action. Whereas, if the central nervous system involved in the reflex action is spinal cord, it is called spinal reflex action. So anyhow, all reflex actions are maintained by an element of central nervous system. And only after that incident or that event had occurred, we come to know that it has happened. At the time of action, we are unaware. So that is the character feature of reflex action. Now, we'll be passing on to sensors. See, organs which are all capable of understanding stimuli, receiving them, and convert them into signals that will be sent to the central nervous system for processing. They are called sensory organs. What type of sense they are all making? Based upon that, we'll be calling them by different names. For example, gustatorreceptors, taste receptors. Okay. Olfactoreceptors, smell. Tangoreceptors, touch. Chemoreceptors, chemicals. Photoreceptors, light. Eye. We are studying that eye as a classical example. Okay. We are having a number of receptors. Olfactoreceptors, which are concerned with smell, is present in the nasal chamber. In that nasal chamber, you are having olfactory epithelium that consists of 3 types of cells. The neurons of the olfactory epithelium extend from the outside environment directly into a pair of broad bean-shaped organs called olfactory bulb, which are the extensions of the limbic system. So, they are concerned with smell. They are olfactoreceptors. Then, nose and touch detect the dissolved chemicals. They are called chemoreceptors. But the difference is that tongue is helping in detecting the taste, gustatorreceptors, whereas smell is with the help of olfactory chambers. Tongue is having taste buds, which will be basically called gustatorreceptors or gustatory receptors. So understand that olfactoreceptors and gustatoreceptors are basically chemoreceptors because it is with the help of chemicals they will perceive the taste or the smell as case may be. And our tongue is mainly, they are also present in other parts of the buccal cavity, in the epithelium of the buccal cavity, but mainly in the tongue, you are having the taste receptors, and I is the olfactory. The I is the receptor for light. So visual reception is with the help of the eyes. A pair of eyes located on the orbit and are coordinated in their action are present. Each eye is a spherical ball, hollow ball, having 3 layers. From outside, they are called the sclera, the middle choroid, and inner retina. Sclera is the outer tough fibrous covering, which is bulged out in the form of a convex structure anteriorly. And it is this part that you will be seeing. This bulged out anterior part is transparent. This is called cornea. Cornea is without blood supply. The second layer is highly vascularized, pigmented layer called the choroid layer. It is ending up at the point where the cornea bulges out, and it forms a thickened structure called ciliary body. This ciliary body is having 2 structures starting from it. One, it is having a curtain-like structure called iris, which is the black of our eye. Okay, this iris is a curtain-like fold having a central opening called pupil, through which light will be entering. And second one is the ciliary body is having certain specialized muscles called ciliary muscles. These ciliary muscles are having a property of involuntary contraction and dilation. It is under the autonomous nervous system, and starting from that you are having a suspensory ligament. Suspensory ligament holds a crystalline biconvex lens in position, which is diagonally opposite to the cornea and pupil. It is this lens that tries to focus light on the retina, which is the innermost layer. Okay. The innermost layer called the retina is having 3 layers of cells. What are they? On the outermost, outermost means which is very close to the choroid layer, you are having photosensitive layer. Middle, bipolar cells, and the inner, ganglia cells. So, this is the structure of retina. Retina is the photosensitive layer that is present only in the posterior most part of the eyeball, which is having 3 layers of cells. Photosensitive cells are of 2 types, rods and cones. These rods are elongated structures which are all helping in see in dim light. In dim light. So it's called scotopic vision, black and white. Image will be alone form. Whereas cones are giving bright images. So only in very bright light images will be formed, but it will be giving color vision. So it's called photopic, only sensitive to bright light. Cones are more concentrated to the posterior most part, where you are having a yellow circular area called macula lutea. Macula lutea is a yellow spot, at the center of which you are having a thinned out area called fovea centralis. In fovea, you are having only cones. So if image is formed here, light is falling on here, you will be having the brightest vision, cleanest vision. Okay. And just behind that or below that, you are having another spot called blind spot. Why it is called so? Because here rods and cones are absent. So if light is falling on that, there will be no vision. So it is called blind spot. It is through this spot that the optic nerves are leaving the eye and blood vessels are entering the eye. So this spot called blind spot is having no rods and cones. Fovea having only cones. In other regions, more rods are present than cones. Okay. Thus, the cavity of the eye is divided into two chambers. A small chamber between cornea and the lens called aqueous chamber, which is filled with a fluid secreted by the ciliary body called aqueous humor that nourishes both lens and cornea. Remember, cornea and the lens are having no blood supply. This is the reason why cornea of any person can be transplanted to any other person. The second chamber is the wide chamber between the lens and the retina. It is called vitreous chamber, which is filled with a jelly-like fluid called vitreous humor, which is responsible for maintaining the shape of the eyeball, so that light will be always falling on the retina correctly. Now, how the eye is capable of making images and vision, let us study that part. When light is coming to the eyes, it is focused with the convexity of the cornea and by the lens, so that it is falling on the photosensitive after passing through ganglia, bipolar and photosensitive layer. So light first touches the ganglia. Our retina is said to be an inverted retina because light is not first touching on the photosensitive. Photosensitive part is on the outermost part. Light is first touching on the ganglia cells. Now, when light falls on rods and cones, there occur a change in permeability. And that change in permeability causes a signal. A change in permeability causes a signal which is traveling to the bipolar cells and then reaching the ganglia cells. Now the ganglia cells will be producing action potential which is carried through the optic nerve to the brain. How the rods and cones are capable of detecting light and producing signal. They are having a pigment called rhodopsin and iodopsin in them. When light fall on them, that pigment part, retinol and opsin, a protein, these two become separated. That pigment is having two parts. A retinol, which is a derivative of carotene, vitamin A. This is why we say that vitamin A is essential for clear sight. That vitamin A is converted to prokaryotin, is converted into the retinol. That retinol and the opsin protein are associated in a particular way. When light fall on them, the opsin and retinol dissociate from each other. And that dissociation is responsible for a changing permeability. Clear? The message is brought to the occipital part of the brain, which conceives it as, or which understands it as vision. So the color, shape, etc. are perceived with the occipital segment of the brain. This is how vision is caused. Okay, opsin and retinol. This retinol is an aldehyde of vitamin A. And the opsin is a protein that becomes dissociated. Potential differences are generated in the photoreceptor cells that produce a signal that generate action potential in the ganglion cells after passing through the bipolar cells. These are all reaching the visual cortex of the area of the brain that will be recognizing them, and vision is thus caused. Clear? Now coming to the ear. So eye is the photoreceptor, whereas ears are two in one. It is statoacoustic, both balance and hearing. Mammalian ear are characterized by the presence of an external ear part, which consists of a pinna and an auditory canal. At the base of this auditory canal, you are having the tympanic membrane. We are having external ear, middle ear, internal ear. The middle ear is a cavity within the bone which is connected to the pharynx through the eustachian tube. This eustachian tube will be helping in maintaining the air pressure both outside and inside same. The tympanum is called the eardrum. It is a membrane which is formed up of the mucal membrane covered with skin externally and mucal membrane on the inside. Okay, we are having the tympanic membrane composed of connective tissue covered with skin outside and mucous membrane inside. The chamber inside the middle ear is having three ear bones called ear ossicles, which are the smallest bones of the body. That is yet another mammalian character. Malleus, incus, stapes. Malleus is remaining connected with the tympanum. And the smallest bone, stapes, is in connection with a opening in the middle, in the internal ear called the oval window, fenestra ovalis. Now, the inner ear is made up of a highly specialized, intricately coiled, a characteristic shaped membrane filled with fluid. That is called membranous labyrinth, which is filled with the endolymph. That membrane is filled with the fluid called endolymph. It is placed in a similarly shaped cavity tunneled in the bone. So it's called bony labyrinth. That bony labyrinth is filled with another fluid called perilymph. So in the fluid filled membrane bony labyrinth, the fluid filled membranous labyrinth is, that is the structure of the inner ear. This membranous labyrinth has two parts. A lower part called cochlea, which is concerned with hearing, and an upper vestibular organs, which is concerned with balancing. Now, coming to the lower part, cochlea is a spirally coiled tubular structure whose cavity is filled with fluid. We are having the upper membrane of the membranous labyrinth called racemase membrane and the lower membrane called basilar membrane. This is the racemase. This is the bone. Okay. The entire cavity is filled with the fluids, and the space between racemase membrane and the bone is called scala vestibuli. Scala vestibuli is filled with the perilymph. Similarly, the lower membrane is the basilar membrane, and the space between this membrane and the bone is scala tympani, which is also filled with perilymph. Scala vestibuli is beginning with the oval window. Okay, And so vibration which are falling on the tympanum will cause the ear ossicles to vibrate, and these vibrations are related to the oval window, and that causes the fluid in the scala vestibular, that is the perilimb, to make ripples. These ripples are traveling to the very end of that coil, and there is an opening called helicoderma through which they will be coming back. Okay. So this is the membranous labyrinth. Here you are having so this fluid, this stapes, this will cause the fluid here to vibrate. It will be conducted to the very tip, and through the helicoderma it is coming to the lower chamber called scala tympani. Larger on that lower membrane called basilar membrane, there is a characteristic organ called organ of Corti, which consists of sensory cells arranged in a definite fashion, which are all supported by supporting cells. And these sensory cells are having sensory hairs, which are having stereocilia, which are remaining in touch with the a membranous structure called tectoral membrane. The organ of Corti is resting on basilar membrane. When the fluid in the scala tympani is vibrating, that will cause a fluctuation, and that will cause the organ of Corti to move up and down, so that the stereocilia, which are remaining in touch with the tectoral membrane, undergoes bending to different degrees, and that bending will cause stimulation in the sensory cells that will cause an action potential in them, and these action potentials are carried to the 8th cranial nerve, auditory nerve, that will be perceiving as sound. They will be taken to the temporal region, which will be perceiving them as sound. Okay, so this is the sound hearing part, namely the organ of Corti. Now, the upper part, called vestibular part, consists of, the vestibular part consists of two regions. One, an otolith part and a semicircular canal. We are having an otolith part, which is having an upper utricle and a lower saccule, and three semicircular canals. These three semicircular canals are placed in three different planes, right angle to each other. And these semicircular canals are all having a ampulla at one end. What is an ampulla? Ampulla means a swelling. See, just like this. These semicircular canals open and begin both into the cavity of the vestibular part, okay, to the vestibular apparatus. This swelling called ampulla is having a specialized organ called crista ampullaris. Crista ampullaris is concerned with our angular equilibrium or dynamic equilibrium. That is, when our body is moving at an angle or when your body is showing any type of movement, the fluid inside that semicircular canal will be moving, and these movements are understood by the crista ambularis, which sends this message to the brain, which will be understanding in which direction your body is moving, and equilibrium will be maintained by the brain. Similarly, the upper utriculus and saccule, which are both called otolith, will be having another thickened structure called macula. This macula is bringing about the linear equilibrium or static equilibrium, meaning even when you are all standing without any movement, or if you are all moving in a straight line without any tilt to any side, even then your body understands and maintain your equilibrium. That is with the help of the macula present within the saccule and utricle. So saccule and utricle together is called otolith, and the three semicircular canal together forms the upper part, which is called the vestibular part. Okay. This vestibular part is concerned with equilibrium. Now, the middle ear, we are having 3 ear ossicles. These 3 ear ossicles are not only mere helping in transmission of transmission of these vibrations from tympanum to the oval window, but they are also amplifying the sound vibrations. Because of their leverage action, they can amplify the effect of vibrations. Okay, so middle ear is not only simply conducting, but it also acts as a built-in amplifier. That's why our ears are so sensitive. This is why we say that our ears are both stato and acoustic in function, both helping in maintaining equilibrium and in hearing. The external ear receives sound waves and direct them to the eardrum. Eardrum vibrates in response to the sound waves, and these will be transmitted through the ear ossicles to the oval window. Vibrations are passed through the oval window on to the fluid in the cochlea, where they generate waves in the limbs. The wave in the limb induces a ripple in the basilar membrane, and the movement of the basilar membrane bends the hair cells present in them, that is in the organ of Corti, against the tectoral membrane. As a result, nerve impulses are generated in the associated afferent neuron. These impulses are transmitted by the afferent fibers via auditory nerve to the auditory cortex of the brain. That's how we will be recognizing this sound. So with that, we come to the conclusion of the discussion of this chapter, and we'll be saying goodbye for the time being, and we'll be meeting again with the next chapter next time. Until then, goodbye and take care, all of you. So thank you all. Thank you very much.